empiezas dando un ejemplo. Sí, lo que entonces, luego.
One minute. Is the microphone working? No, no, only like this. I'll hand over to each. I'm sorry. This is how it is. All right. Good morning to you all. We have a busy agenda and we have a lot to get to, so let's get started. Good morning to you all. On behalf of Pensoft Publishers, LifeWatch ERIC, and the Naturalis Biodiversity Center, I welcome you to the first session of our joint symposium. To both those of you in the room and those attending online, we thank you for your participation and we hope you find the agenda we've prepared meaningful and engaging. We seem to be, can I have the next slide? Yeah. It was working a minute ago, but there we go. So my name is Peter Buzakov. I'm a science communications expert for Pensoft Publishers. Before anything else, I'd like to thank our two collaborators in this endeavor, Christos Arvanitidis from the LifeWatch ERIC and Nios Ras from the Naturalis Biodiversity Center. This symposium is the latest chapter of a long collaboration between our three institutions, and we're very grateful for all their support. Today, can I have the next slide? Yes. Today we will be exploring three main themes with an array of diverse and interesting research projects and initiatives: knowledge transfer, research continuity, and the reusability of scientific results. Our own work at Pensoft has shown us time and time again just how important the legacy of these projects are, the legacy of their results. Otherwise, the impact they generate would only be felt within the temporal bounds of the initiatives that gave rise to them to begin with. So, how will we explore this? Well, there are two sessions to this topic. One is today, one is tomorrow. Same room, same time. You're welcome to join us again. We have 16 abstracts in total, featuring more than 90 authors, more than 20 research projects and initiatives, and more than 30 institutions and organizations. We have three main goals with the symposium. First, we want to showcase the importance of open data to the long-term prominence of research results. Second, we want to link targeted communication to actionable knowledge in this regard. And last but not least, we want to explore the pathways to impact for these scientific outputs in relation to decision-making. I take this opportunity to say a few words on Pensoft. Some of us met us already at the booth, but nevertheless, we are a company of more than 30 years based out of Sofia, Bulgaria. We specialize in academic publishing and science communication. In terms of the former, we stand behind more than 80 academic journals and more than 1,000 scientific books. We're also behind the ARFA publishing platform. Oh, sorry, you switched the slide, I think. Yeah, thank you. We also stand behind the ARFA publishing platform, a full-featured open access platform. The second iteration of our proprietary ARFA writing tool will be showcased later today. All of this is to say really that we work to innovate the research landscape in terms of data publication, semantic annotations of data and text. And we also pursue a number of research projects, more than 40 under the Horizon Europe and Horizon 2020 programs. Our specialty in this regard is communication, dissemination, and exploitation, linking the results of these projects to the right audiences, to the relevant stakeholders. Recently, we began sharing our experience in this regard in what we call the Science Radar newsletter, You see the QR code on the screen. If you scan it, you can subscribe to this newsletter. We like to showcase the importance of targeted science communication and data innovation, but not only from our own portfolio. So there is an email address here, and we'd love to hear from you, your inputs, your innovations, your stories, your experiences, and we'd love to include them in the next issue. So please feel free to do that. Last but not least, we are also exhibitors at Living Data. As many of you already saw us at booth number 9, we have an array of materials to share with you, and we'd love to continue the conversation started during this symposium, and you can find out more about the numerous PenSoft-affiliated projects you'll hear about today. Now, before I continue, a couple of housekeeping rules for the session. We'll have 8 talks of a 10-minute duration. I kindly ask our speakers to keep to the time limit so that everyone gets their due. 1 minute before the end, I will subtly indicate to the speaker that their time will be elapsing. And I kindly ask our audience to save their questions and observations for the Q&A segment at the end, as we will have the opportunity to delve a little deeper during that segment. And now, without further ado, I'd like to introduce our first speaker. You already heard his name, Christos Arvanitidis from LifeWatch ERIC, who'll be talking to us about the past, present, and future of what we call the Biodiversity Knowledge Hub. Let me just give you the microphone. Yeah. Here.
Put it in there. Thank you, Mr. Moderator. That was quite a good introduction, and now we have to run to keep up with the time. Well, we're not on the first slide, I'm sure. So again, let me repeat till we go there. The title is Biodiversity Knowledge Hub, Past, Present, and Future. I'll deal with the past and the present, and my colleague Niels Reyes, he will continue with the future because there is a future. This title actually and this activity implies a lot of challenges, the most important of which are that the data and the tools, they are distributed across the research infrastructures, repositories, global aggregators, and so on. The users do not need just data, but they also need actionable workflows, and those workflows, it should be better, to be cross-domain in order to do their job. And we will come back to this in a minute. And finally, the policy needs traceable baselines, evidence-based, robust trends at multiple scales in order to be able to take decisions and manage our environment. The next one shows something about the bicycle project. The Biodiversity Knowledge Hub was a product, the central research product of the bicycle project funded by the EC and ended last year. There were a lot of things that have been achieved. You see, the most important on this slide, but I will stress on 2. The first is that the fair biodiversity data work across the research infrastructures, repositories, and expert communities can be integrated. That's one of the best things we were able, to prove. And the last one, which says that biodiversity turns fragmented the BKH turns the fragmented biodiversity assets into dynamically linked, living, interoperable knowledge ready for science and policy, and perhaps for many other purposes. And if you want me to give you a definition of what is the Biodiversity Knowledge Hub, I can give you many angles, which are already shown on this slide. But I would stress on the first one, which is, the one-stop portal for understanding the complex but increasingly interconnected landscape of biodiversity research infrastructures in Europe and internationally. And those infrastructures actually, they shoulder responsibility for the maintenance of those services and the data. And it's a place where virtual research environments are formed from individual modular services. So it's a dynamic environment there. Can we play the video now, please? We're going to see, a video. We're going to watch a video which shows how the people, how the users can access the Biodiversity Knowledge Hub and can explore it. And yeah.
This is the Biodiversity Knowledge Hub, the major outcome of a 3-year project funded by the European Commission that saw 14 key institutions from 10 European countries come together to devise a knowledge broker for the extremely diverse realm of biodiversity data. While building on their own data infrastructures and the two-way links between them, the bicycle partners shared tons of experience and know-how with each other. They all agreed, researchers, taxonomists, collection managers, informaticians, publishers, and even research infrastructures have been struggling to fully grasp the biodiversity data landscape, complete with the related guidelines, tools, and services. So, the Biodiversity Knowledge Hub was designed to be the stepping point in one's work whenever biodiversity data are concerned. Researchers, for example, can use the Biodiversity Knowledge Hub to familiarise themselves with the data types relevant to their work, identify the sources where they need to search for them, and then learn how to use them, so their studies comply with the best linked and fair research practices. Meanwhile, taxonomists will figure out how to easily locate and access various checklists, taxonomic treatments.
Thank you. Can we go back to the slides, please, and continue, actually? We'll continue with this video by the end of the session and during the question and answers as well with another one. So can we go back to the slide we were a minute before? Yeah. Can I continue? Yes, I can continue. No. One back. Fantastic. The strategic product in Biodiversity Knowledge Hub was the FAIR Data Place. And the FAIR Data Place is here located at the intersection between the 4 scientific domains. We have taxon names and OTUs. We have specimens and samples. We have sequences and genomes, and finally life cycles literature. So the interconnection of all these things liberates data that are interlinked and well searched. Now, how difficult is for this data to be harmonized, to be cross-searched, and to be finally managed? It can be shown, in this on this slide, where we have, from the ELTER, those 5 system spheres, each of which requires and generates a lot of different types of data. And that data, actually, they need, to be harmonized and managed properly in order to be finally analyzed under this, let's say, whole system approach in order to get the results and their interpretation. These were kindly donated by our colleague, Michael Myrtle, the coordinator of the ELTER research infrastructure. And now just a couple of slides to show which data, which services we support through the Biodiversity Knowledge Hub. For example, from Sibelius, we have the Sparkle endpoint. We have from Disco the Discover. From Elixir, we have the ENA cross-reference search, and we have, the notebooks. From the Zenodo, we have the Biodiversity Literature Repository. And it continues here, the Biodiversity BMC. From LiveWatch, we have the LiveBlog. From Plazi, we have the Treatments Bank. And finally, from Pensoft, we have the alpha writing tool and also the nano publications. But please, these are just examples. There are many more in there. And going ahead, we should have a look to this slide, which shows, how the users can access the BK8s and also can explore the resources. And allow me to give you just one one of the resources here, which is explore the linked data. It's a functionality in our website where the people, the users can search in the data sets and the databases of up to now 17 global aggregators and research infrastructures and repositories, etc., such organizations. And the number is growing as we go by because we have the responsibility, as I said, to maintain these services. The final slide from me is about the benefits for other research infrastructures to join the BKH, the Biodiversity Knowledge Hub. So there is an open call, if you like, for all of you, if you represent, research infrastructures. I think that the most important benefits is the first one, the exposure and visibility for their tools and their services. And the last one, the increased use and efficiency of investments in data aggregation and management. And now it's your turn.
4 minutes? Okay, good.
This one goes, this one back. If you want, you can use it. Go down like this.
No, it's where?
Yeah.
Okay. Okay, let me move on. So BMD. So building on all this technology and the interoperability, what has been developed on a bicycle, we're now moving forward with BMD. Okay, I'll just use this one. So what's new here? So with BMD, we're going to mainstream high throughput biodiversity monitoring tools and AI-powered species identification as one of the elements. The final slide will show the entire infrastructure that we're going to build. But this is key of BMD. So we have heard of the keynote of Alice. There's a huge data gap. Data is sparse. And what we're trying to do here is to fill that by high throughput monitoring. So we're going to from cameras with 4G connection and then all the way to automated species identification and then mobilizing all these data automatically to GBIF. In a standardized way, and then pick up these data from GBIF in virtual research environments, similar technologies as has been developed on the Biodiversity Knowledge Hub. So key here is to do trend analysis, that we start mobilizing historical baseline data, which is very essential to fill the gaps. So together, the historical baseline data, together with the high throughput data, go into GBIF. From there, we're going to pick it up. And then we're going to build and interoperate these systems with the European research infrastructures and the ESFRI infrastructures into the global aggregators. We use open standards and APIs to reuse and federate the data straightforward, so everybody can access this data and should be made publicly available, taking into account the sensitive species. So we're not going to make that available at high resolution. We have solutions for that, but that takes too much this presentation. So now data generation in itself is not a means by itself. So we need to analyze this data. We're building this infrastructure for the managers of Natura 2000 in Europe. So this, well, we're feeding into the monitoring needs of the protected species across Europe. We have 27,000 Natura 2000 sites across the terrestrial, freshwater, and marine realms, and we're building tools to analyze these data together with those stakeholders. So, and that's the power of BMD. This is not a scientific endeavor, it's an innovation action, and we're bringing this technology to the end users in the communities. So we cover all three realms. The users actually define the analysis. We're building on Natura 2000 designation reports and national checklists, and all this data will be mobilized and made available into GBIF. And then we're coming to the total infrastructure. This is what we're going to deliver. Central there is the single access point. We have the stakeholders. Very important is that everything what we do is fair, so the input, the output, but also the workflows. So we're building a workflow hub and RO crates, so everything can be reproduced at a later stage. And we're keeping the workflow together with the input data from GBIF and the end product, and from there you can reproduce it at any moment in time. And then, well, the power here is that we have all these different elements. So the plug and play, the mobilization of historical baseline data. We have a data catalog with spatial data and drivers of change. Then we have a GIS viewer just to visualize these data, and making the tools available to analyze this data. And this is brought together in a single access point so that the end users always know where to find the data. And well, we're building on developing these endpoints similar to the Biodiversity Knowledge Hub. So that will be integrated there. And I think that will be my last slide. Yes, thank you.
Thank you to Christos and Niels. It was a very inspiring presentation to kick off on. I'd now like to invite Quentin Groom with a showcase of another duo of project, BCube and OneStop.
Yes, I should have worn a collar.
You can use your badge or your...
Okay. Hi, everyone. So I'm Quentin Groom from Miser Botanic Garden, and some of my collaborators here are in the room. I see Peter de Smet there, and I see Demiano over there, and maybe there's some others. And I'm going to talk about how we've tried to make sure the products of our research project have some longer life and better use for many people. So I will try looking forward. Good. So a lot of the stuff I'm going to talk about today, you all know already. The ephemeral nature of projects. They're short, but we want to have everything to have a longer lifespan. We're all driven by grants, narrow scopes as well. But of course, as soon as they've finished, there's a strong possibility that the outputs lose visibility, that people don't pick them up and they're not used. And that's part of what I'm trying to resolve. What we have has value, and we need to be thinking about that value during all the projects. But we have this issue within a project which has to be internally consistent, and that all the aspects and bits and pieces of that project need to work together as that project. But that's a conflict between that and the wider landscape of other projects going on, infrastructures going on, and being compatible with all of that too. We also have this conflict in projects that we need to deliver fast, compared to sustainable things, which often take a lot longer. And so there's a constant fight, not a fight literally, but a tension within projects on that as well. So my first venture into coordinating a project was the Trias project, which several of you were involved in. That was to do with tracking invasive alien species. I'm interested in invasive species, both from a sort of socio-political point of view and from a biogeographical point of view as well. And Trias were very much about workflows, making open workflows. It's something that my team have been involved in, but also Peter de Smet and Demiano have been very involved in trying to make workflows that are open, transferable, and repeatable as well. And those workflows are now being picked up in other projects that I coordinate, BCube and OneStop. But of course, that's kind of inevitable because it's some of the same partners within there. We obviously want to reuse stuff we've made in the past. The challenge for us is, can we get other people outside our projects to pick up on those things as well and get those data and those workflows reused by other people? So I'm quite pleased that the Guardius project, which is a sister project to OneStop, is using a workflow that we created. Amy Davis, I'll talk a little bit about local wisdom about species distribution modeling. But also, yeah, we need to figure out ways that we can maintain those workflows after the project finishes and get more of a bigger community involved. So to say a little bit about this, all of this is built on other open infrastructures. Infrastructure is super important to us, and oh, shouldn't touch that. Things like GBIF, which is an excellent example of an open infrastructure. Zenodo, which is to some extent a dumping ground of stuff, but incredibly useful for being able to disseminate things and put a DOI on on documents and software. GitHub, it's commercial, but I don't think we should reject it because of that. It's also open, a great way to version software, even documents. And we use things like Wikidata, which is very good for those sort of bringing a different community involved. And I've used it personally for things like biographies of people and data on people a lot. And so it's also part of that infrastructure. We also build everything on common global standards. So standards are being agreed by the community. It takes a long time to work on standards, but because it's worked on by a community, it means it's a stronger foundation on what to build. And we design for reuse, which takes time. It doesn't reuse, and design for that doesn't show much instant impact, but for the longer term impact, it's essential. So this map here is just an example of the output from the Wisdom workflow, for example. It was written in R a few years ago. You can see it was published in 2024, but it was actually written in the years before that. It's an ensemble modeling workflow, but it now needs updating. R tends to age quite quickly in terms of the packages and the dates on the packages. And so that work is being done within OneStop and within Guardius as well to keep that alive. And thinking about how we can do that and maintain that long term is difficult. So communities, I mentioned it already. I think without communities around projects, the community helps maintain the software and the data, but also the community is part of that co-creational aspect to it, because the community tells the people doing the coding what they want. So this was a slide I put together. I don't know if there are any science funders in the room, probably not, but it would be lovely to have a few here. But this was a slide for them. So yes, this incentivizing novelty over longevity. Yeah, I mean, it's a mixed picture amongst funders, but it's rare that they don't want any novelty in a project, or that they go out to try and maintain things or keep a project going for longer or extend things. And so, yeah, infrastructure. I was thinking actually when I was looking out the window there and seeing all the cars go past, if you have infrastructure like that road out there, when it's broken, you don't throw away the whole road, you fix the road. And that's how you should be thinking of infrastructure, is not in terms of sort of picking it up and putting it down again when you don't want it anymore, or the emphasis is changed like that. So funders making sure that every project thinks of the sustainability of its output are very important. And yeah, don't fund projects just to produce a bunch of papers, but also think about how the data and the software used don't just become obsolescent and lost afterwards. And we have some aspects of that. I can't blame funders completely. It's down to scientists too. And so we have this conflict between, on the one hand, we have all these project outputs, we want to move on forward and things like this, and there's a constant tussle between the two. Often when people retire in the Botanic Garden, they come to us in the data team and ask, well, what can I do with my data? Well, they should have done that 10 years ago. Their data has now become old. They've just suddenly realized that their legacy is sat in a notebook somewhere. And that's a bit sad, and we can't always resurrect those things. It would have been much better had they done it 10 years ago when they could have got some value out of that. And that's what I keep telling. And yeah, so they want to have a legacy, and we all want to have a legacy, and we all want to have impact, and we can only do that if we share. So I think I've said all of this. Yeah, I will skip that because I'm pretty sure. Yeah. Okay. So I just want to quickly mention the One Stop project because they're actually funding me to stand in this spot. That's a project to coordinate. It's tried to be the full one-stop shop for data management within invasive species. We have a pillar on detection that's doing AI methods to detect invasive species, eDNA detect invasive species. We have a pillar on prioritization, trying to make sure that people who are managing invasive species know how to prioritize the time and money that they get for it, which is very limited onto what is most effective on managing invasive species. The dissemination is about the dissemination of things like the detections. So we want to push all of our data, we collect all those detections to GBIF, and then we want to alert people who need to know about the invasive species on land. And of course, the human aspect of invasive species, which in terms of biology is very important in invasion biology. Why do people bring these species to their country in the first place? What policies do we need? And also what societal changes do we need to reduce invasive species from that perspective? And one of the bits of reuse in this project is there was a project called Riparius, which I wasn't involved in. I think Peter was involved in that. But it produced an alert system for Belgium. One Stop is now taking that code, extending it to the whole of Europe. It's based on GBIF, so anything that gets published for Europe can be seen on this and will alert people in emails or on the website to whether that species is sort of popping up in their country, what have you, and it's very configurable as well. So I think that's it. Hopefully I'll get in time. Oh, that's great.
Thank you very much, Quentin. Certainly a lot to reflect on how we conduct our projects, how we pursue our objectives, and I'm sure this will be the subject of extensive discussion. Our next speaker is also for the Meissen Botanic Garden. I'd like to welcome Patricia Mergen. She'll be speaking to us about nomenclature and how we'll be bringing it back into the lamplight.
Is this working? Okay. So good morning, everyone, or another time for the colleagues online. So I'm presenting on behalf of the first author, Eli Saliba. He couldn't be here, but he's following online. So put anything in the chat if I forget something. So where this idea comes from is because in the TEDWIC Biodiversity Information Standard Group, we decided already a few years ago to have subcommittees of important topics that should help the executive committee. And one of these groups is the subcommittee on fundraising and partnership. So here I will speak more about the partnership part, although I know most of you would prefer the fundraising, I guess. But I have the vice chair, Eli Saliba, who is from GB France, who is helping in this group. And here you can see the common denominator. He is also part of the commission of the International Zoology Nomenclature, the ICZN, together with some known figures of the commission like Thomas Papp or Richard Pyle, who have historically also been very long active in TEDWIC and in the different working groups. So we decided that we should, as first try, do collaboration with them as to collaborate. So first, we looked at some definitions because from the literature we see that there is often a confusion what is taxonomy, which by definition is just a classification of objects. Originally, it was for organisms, but nowadays, if you Google, you can find taxonomies of anything, laws, politics, thing, trucks, whatever, and they all call it taxonomy in the term of classification, while nomenclature is more conventions and rules on how to form the names that can be informal or very formal and structured with standards and principles, like it is the case in the Commission about biological nomenclature. So in May 25, after some discussions and small workshops we did when Ali and a colleague of him came to the Meise Botanic Garden, we had several online meetings with Rich and Thomas Papp, and we ended up having an MOU signed with these three main goals: to have the ICZN more actively engaged in the TEDWIC community, that it's actually the case, and also help in the different working groups. And TEDWIC will also see with its standard how it can facilitate the work of the ICZN representatives. And we will also see that with this first example, we can continue and make also MOUs with other codes in botany or in microorganism and so on. And also, of course, to see how we can work with Zoobank and other taxonomic journals. So we also digged a bit into the history, where we found that ICZN back in 2006 was actually a member of TEDWIC and was also an associated partner to GBIF, but over the years they were not anymore officially members, but there was still a lot of commissioners involved when we discussed with them, because they are a bit overwhelmed by work or also economical issue. They found the MOU a good compromise to bring this back and to formalize again the visibility and the collaboration between these two important institutions. And we hope that it will be a win-win situation. So in practice, we thought one activity could be to see how we can speed up the description and revision of species. So from the literature in 2012, they saw that it could take up to 21 years or more from the discovery of the species to its publication, officially valid. And we find also in the literature that there is a big variation between the taxon. Like with genetics, it can speed up things quite a lot, while for others it still takes quite some years from the discovery to the official publication. So one first activity we want to do and try is to see if there can be a collaboration to have ready-to-use guidelines to assist scientists who are not so familiar with the nomenclature rules, because there is a long learning curve that they can use to publish more rapidly in the taxonomic papers. And we think it's also important to organize trainings on the nomenclature for biodiversity informatics, because that's a bit forgotten, and also we saw via other projects that it's not something really priority in the official curricula anymore in universities or so. And so that we can bring somehow nomenclature, classical taxonomy, again in joint projects to achieve that we can publish more rapidly new species or revisions. And we will, as I said, try to have similar agreements with other existing codes. So that's, I just want to really thank the Commission and TEDWIC because we passed a lot of time discussing about this subject that is not so easy and where there are many differing opinions. And I also want to thank the different funders that enabled that I could be here, which also show that economically it's not so easy, because I needed 4 different sources to be able to be here as a speaker physically. So I thank you a lot for your attention and look forward to your questions during the discussion session.
Thank you, Patricia. Very useful. I certainly learned a lot, and I had some misconceptions on the topic. We carry on with the theme of taxonomy with David Fichtmüller of the Botanic Garden and Botanical Museum of Germany.
Yes. Hello, everybody. So I'm going to show you the different side, the side of actually reusing data from different projects. I'm not going to read the mouthful of a title over there. So what I'm going to show you today is what I've worked on in the past years, and in particular, the problems I encountered along the way when reusing data from other projects. And well, basically, I'm going to tell you why data quality matters for data reuse. It's not really surprising, but still quite an important topic in this context. So, but before we begin, I want to point out some caveats, some things to keep in mind when I talk about this. I'm mostly focusing on person names coming from GBIF. And with that, I use a lot of generalization and heuristics to process them. There's a really great article, Falsehood Programmers Believe About Names. I pretty much touched on every of those falsehoods. I'm aware of that. And also, I have quite a Eurocentric view for this processing. You will see later why. But nonetheless, the conclusion I'm going to present is still valid, even if you take all of this into account. Now, let's take a look. During the Tetris project, which I'm employed on, we had the goal to systematically determine the taxonomic expertise in Europe from different sources with focus on different taxonomic groups and countries. That's quite a wide sentence there. Let's look at some of the and I focused on specimen data. With that, now, next question, well, who is an expert in that context? Well, for my analysis, an expert is anybody who has either collected or identified a specimen. Where does data come from? I already mentioned it. It's from GBIF. And what's the time frame here? Well, we thought for an up-to-date view on like what is our current expertise, it would be useful to limit it to the past 10 years. Wouldn't really help much to know that which expertise we had 100 years ago. And now the tricky question, what is Europe? Well, in this context, we decided to go with a really wide definition of the European political community, because the goal is of this analysis to influence policymaking. We decided to go with that, which is pretty much anything you can define within the broader sense of Europe, with the exception of Russia and Belarus. So this is just a group that formed during the after the invasion of Ukraine, and this we thought is a good boundary definition for us. So then, who is a European expert? Well, any expert that comes from Europe or works in a European institution. Here's the workflow we came up with. Well, we first download the data, we clean the names, we do some matching with ORCIDs, we then get the institutions to those people, and from those institutions, we get the countries, but also we get the countries of the people themselves, combine that, filter, aggregate by country and taxonomic group, combine it with the data from the taxonomic publication analysis, and in the end, we generate nice maps and charts and analysis. Today, I'm only focusing on the first part, because that's the one that actually relevant to the reuse question here. And so for the download, I was able to use the new GBIF SQL download API, which really, really helped us because it made things so much easier. We ended up with a little over 20 million records. And yeah, we took those records. So that's a classic GBIF record, record by ID, event date, identified by ID, and identification date. Or we split that up, renamed the columns to align them, added the event type, recorded and identified, then able to merge them, got a 28 million records there, and then aggregated them by the name and ID combination. So a little over 500,000 records. Now, that brings us to the first problem. As you saw on the previous slide, there were some that don't have the event by ID field filled out. Question to the room. Of those 517,000 names, how many of them do you think have an event by ID filled out? So any kind of identifier, usually ORCID or Wikidata or some of the others. Keep in mind, it's data from the last 10 years. ORCID has been around for 13 years now. Any guesses, any numbers? Huh? 10, 9, something, well, it's 2% actually. So that's a bit disappointing when you want to reuse the data and have an easy way to match this. So there's no way around to actually match the names to external sources. And now we look at what we have there in event by. Well, different ways of spelling with different, quite often enumerations of people, some using the pipe symbol. That's the recommended way to do it in the Darwin Core Standard. Sometimes abbreviated, sometimes family name and given name in reverse order. But those are all good cases. We can work with that. It's getting a little bit more tricky here. We have institutions. We have the French notation where the family name is written in all caps. We have cases where you have family name and first name, and it's not really clear. So if you know it from context, from language, you know in that case, Nina Müller, it's a German name, Müller is the second name. We have couples where we have only one mention of the family name. And then we have the really tricky cases where it's really hard to come up with proper rules to process them. My personal hate element being the using the same character for the separation between family name and given name as for the enumeration. So you can imagine it's really hard to parse those and split them up. Well, I did some initial cleaning with OpenRefine and then used a Python script for splitting and separating them. And I focused on the most common name types. As you can imagine, with those so many names, it's always hard to match all of the cases. So after cleanup, I ended up with about a little under 500,000 names. And well, that script that does the processing can always be improved, but it's like marginal gains. And eventually it's a question, okay, how much time is it worth spending for a case that has, of course, like 20 or 30 times within the script? Then the matching of the data, well, we have GBIF, that's the one where we have the ORCID from the original data, but also from different rows. So if the same person name has an ORCID in one row, and the same name appears in a different row, we could use that. Bionomia, we have the connections to the individual specimen. That's really useful. Wikidata, I wrote a query that to get me all of the living biologists by any sub-job type of biologist. So we were able to match against that. We could use the output of the taxonomic application analysis. So we know people are active, but maybe with that we can find some additional taxonomic groups. And I decided against using the search for ORCID because this will just turn out too much other results that are not relevant to our field. So that brings me to the lessons learned. Well, data from multiple sources is always messy. Identifiers are really useful when they are there. Conventions are really useful when they are used. And human cleaning works quite well, because as a human you can distinguish them quite good, but it doesn't scale. So on a just rough estimate, the number of results we have there, if you spend 10 seconds on each name, and that's not realistic, that's like a low-end level, 10 seconds on each name, and you want to process them all by hand, you will need 1 year of full-time work, 40 hours a week, with a bit of vacation. And Well, that sounds like an AI solvable task. And I tried to apply to a certain point, but it ended up that at least ChatGPT in the background just wrote a Python script doing applying the rules that I told them. So that wasn't the magical solution I had hoped for. There are probably better ways, but I wasn't able to do that. I'd be interested to hear your thoughts on that. So how do we get to a better future? Well, it requires involvement of all the people here. So if you're a researcher, get an ORCID and use it whenever possible, and follow conventions for data formats. If you are an aggregator or working for an aggregator, flag data quality issues early on and really incentivize people to improve their data quality, ideally before it hits the network, because updating things later on is always trickier than doing it right in the beginning. If you are a software developer working on systems for data entry, make it easy for people to input consistent data. Make it the easiest way should be the best way, otherwise people will just be lazy and do it in the, whatever fits best. And warn about inconsistencies. So like, oh, we recognize you use different separators here. Maybe use a pipe instead because that's the recommended one. And if you're a reviewer or editor of a journal or data journal, well, check for data quality, especially if it's like hidden in the appendix or reference material, because that's the data that will eventually end up in the data networks. So I have rejected or recommended a paper to do active revisions because the data quality was not up to standard. So my final message, data quality and data consistency are key factors for effective data reuse. And with that, I thank you for your attention. Check out the work by Quentin and his team. And there's the list, the article, False Programmers Believe About Names.
The head, I think. Now, halfway through our session, we move on to an exciting topic, genomics, Fair Data Foundation for Biodiversity Genomics to be specific. And I invite now to the stage Joanna Poperio from the European Bioinformatics Institute.
Hi, good morning, everyone. So I'm Joanna, as Peter graciously presented me, and I work at the EDI, and I'm here to present the work that we've been doing in a project, Biodiversity Genomics Europe, and the work not only developed by me, but by the team that is working in a specific part of this project. So, biodiversity genomics has been increasing in this last few years. So we see a lot of large number of initiatives trying to scale up sequencing and with a mission of accelerating the use of genomic science for biodiversity conservation. In this talk, I'm mostly going to refer to reference libraries, both in reference genomes, so the production of reference genomes, but also in the production of DNA barcoding. And these reference libraries are fundamental for the application of biodiversity monitoring through metabarcoding, population genomics, and other methods. So Biodiversity Genomics Europe is a European project funded by the EC. It's a 3.5 years project, which is nearly finishing. And this project joins two main communities in Europe. The European Reference Genome Atlas. Oh, sorry, I forgot to change this. The European Reference Genome Atlas that Rob's going to talk a little bit more after me, and the IBOL Europe community. So BGE is a project that is trying to coordinate and upscale DNA barcoding, and sequencing of biodiversity in Europe, and also trying to develop synergies to support the communities, but going through all the stages of the process. So from sampling through to lab methods, through to bioinformatics analysis and publication of information. What I'm going to focus today is on the efforts that are being done in aligning these efforts done in these communities towards these fair biodiversity genomics data. So in the project, we have a work package that tries to align these communities and the work done between them. And what part of this work is on aligning best practices from the barcoding and reference genome communities, and building a framework for integrating these future efforts in the biodiversity genomics data infrastructure in Europe. And we've been mostly working in 3 main areas by trying to align data and metadata standards, supporting data brokering from the distributed system of production into the data publication, and enhancing data accessibility and interoperability. There are some challenges in merging these 2 communities, because though they all focus on the production of reference libraries, there are specific technical paths and pipelines that they use. The outputs are different, the solutions found are different to deal with different problems. Though there are a lot of overlaps in sample and metadata connection, there's still a need for alignment, because they favor different fields and different information. And there's a differential usage of the overall infrastructure landscape and a need for better linking between the data resources, which is a common problem overall. So here we can see a representation, the quality is not very great, of the data infrastructure landscape that BGE interacts with. You have, for example, I'm sure if there's a pointer here. Well, anyway, so you have the community that produces the data. We have data brokers like COPO and management platforms like COPO and PlutoF. We have the repositories like INSTC, BOLD systems. There are specific repositories, for example, for permits. There's analysis infrastructures like Ensemble. There's Workflow Hub for supporting pipelines, and then the natural history collection. And you can see that, you can't see very well, but most of these have a straight line. Some of this has a dotted line where there is links that we're trying to implement, but they're not already there. So where did we focus mainly? So we focused on aligning standards and metadata, data linking, what to do with pipelines and workflows, enhancing interoperability with fair digital objects, and also some data management training, which is fundamental if you're running a distributed system of production. So in terms of metadata standards alignment, the European Reference Genome Atlas made a big effort in its beginning to properly define a rich collection of metadata fields that should be collected and published together with the sample information. So they built a sample metadata manifest that then, and that sample metadata manifest that is also based on other biodiversity genomics initiatives like TITL, is then translated or is aligned with the Tree of Life checklist that we have at the ENA. The barcoding community, because most of them deposit their information into BOLD, follows the BOLD metadata standards, and they also took care of building their metadata sheets with the relevant information to add to what was already there. And for metabarcoding, the MixS standards are usually used, which are already represented within INSDC in terms of data publication. So for metadata standards alignment, what we tried to do was align what was being done by the European Reference Genome Atlas and by the barcoding community, and also aligning with further developments that are being done. So iBold or BOLD are developing a barcode core data model, which I think is going to be implemented in BOLD. And there's also the Vibrant Core standards managed by TEDWIC, and the standards, for example, used by the Unite community. So we did some work mapping these the standards that are relevant for BGE using a simple standard for sharing ontology mappings, the SSSOM. So and so we basically what we did is in GitHub, and it is available in case you want to have a look at that. We also did some work in terms of data linking, because we know that linkage is fundamental between research infrastructures. It's always good that you can link your sample to your specimen and to your data. This linkage has been increasing through several efforts, Bicycle one of them that was already mentioned, and other ongoing efforts. And new links have also been created that are relevant for connecting these BGE data sets. One example is the ENA cross-reference search that was already referenced in the Biodiversity Knowledge Hub, where we were able recently to link with the sequence information with the GGBN samples and tissue and samples, and also with BOLD. So this is an ongoing project. We already linked with Unite and Ensemble that are also relevant within the BGE landscape, but this is an ongoing effort that has been increasing, and you can see here an example where this is an example of the BOLD cross-references, and you can see the record and the way the identifiers are linked with each other, and then you could also see the same information that is picked up by GBIF, where you can see the same identifiers being mentioned there. Then in terms of pipelines and workflows, there was an effort being made to put the workflows that are being produced and developed within the project available for reuse by the community. Workflow Hub is a registry that allows describing and sharing these computational workflows. They also allow the submission of SOPs, standard operation procedures now. And we have created within Biodiversity Genomics Europe a collection of workflows and SOPs that are being used by the project and are available for other users. One other thing that we have been working on is trying to increase the usage of fair data objects. And one of these examples is the RO crates. I'm not sure if you're familiar with RO crates, the research object crate, but this is a community effort to try to package information that is collected in a single linked object. So what was done within BGE is that COPO, which is one of our data management platforms that we use, and a broker of data, developed a RO crate profile for the samples that they hold. And that is now available as a possibility. If you want to look at data inside of COPO, you can choose to download in a format, in the RO crate format. One other thing that we have been working on is developing a biodiversity genomics RO crate profile. And the purpose of this profile is to fit the essential metadata that should be stored in a research data object for both reference genomes and barcoding data sets. This is still being in its initial development, so this is under construction, is just something that we have beginning to develop, and we're piloting within BGE to see the acceptance and the usage by the community. Then finally, we are also doing some data management training, as we believe this is fundamental for researchers. We have done a FAIR data management course, and then we integrated some data management lectures within other courses that BGE is organizing within the project. And that's it. Thank you. Oh.
Thank you very much, Joanna. It's very great to see the FAIR principle applied to such a fast-evolving and fascinating field. She already mentioned the ERGA, the European Reference Genome Atlas. So now we'll have a dedicated presentation on it from Robert Waterhouse from the Swiss Institute for Bioinformatics. Before I give him the word, Christos informs me that this is also a laser pointer. Which button do we press? At the back. Okay, if you want to point at something. Here you go.
Okay, thank you very much. I think Joanna has given you a good introduction of some of the outputs from the biodiversity genomics project already. And I also want to thank Quinten for setting up the kind of questions that we want to think about. And this presentation now is an example of community on the one hand and a project on the other. And so I just want to give a few highlights of some of the considerations that we took into account to think about the legacy. So to think about the continuation beyond a 3.5-year European project and where these outputs have a future, if you like. And so the Biodiversity Genomics Europe project is bringing together both the barcoding community and the reference genomes community with partners from across Europe, representing more than 200 different European organizations. With the three kind of overarching goals, on the one hand of building capacity in the production of reference libraries, so reference barcode libraries and reference genomes across Europe. The second goal of upscaling production, so how can we work in a kind of distributed fashion to increase the rate at which we are filling these reference libraries with high quality reference data sets. And the third pillar of demonstrating the use of these reference libraries. So how can they be applied to better understand biodiversity? And so we have the two streams, as alluded to by Joanna earlier. We have the barcoding stream and the genome stream. But really from the outset, we kind of recognize the synergies across these two communities in terms of our overall workflow, which is essentially from the field to the lab, so all the sample collection and processing, through to the sequencing at scale, whether it's reference barcodes or reference genomes, then all the data processing, data analysis, and data deposition steps that have to happen, all the way through to the demonstrating the use of these important reference libraries. On the genome stream side of things, because I'm talking about the European Reference Genome Atlas in this presentation, this is a representation of some of the more fine-grained steps, if you like, along this route from sample acquisition all the way through taxonomic identification, DNA extractions, then into the sequencing machines, out into the assembly process, then down to annotation, so finding the genes in these reference genomes, depositing them in archival repositories, and finally the data analysis at the end. And here, just on the right-hand side, our kind of final product, if you like, which is the reference genome and its annotation for use by the community. Importantly here, though, it's to say that this project was designed to be well connected and aligned with our two community efforts. So Eyeball Europe on the barcoding side of things and ERGA on the reference genome side of things. So we have the two streams, the barcoding stream and the genome stream, being connected to the communities through the Biodiversity Genomics Europe project. And this involves quite a lot of different things. I will focus mainly on some of the more sort of tangible outputs, if you like, but it's also about supporting the communities in terms of their secretariat, their executive boards, their governance, etc., but also the community membership. It also has been really important two-way kind of dialogue in communications between the project and the communities to raise the profile of both and how we interact. Another kind of key output, if you like, is the ability to support and manage and run these research webinar series. So being run through the project, but being open to the entire community, so bringing people together. And then the ones I'm going to focus on are these tools, these resources, the development of best practices, platforms, etc., during the lifetime of the project that can then be ultimately taken up and used by the two communities, in our case, the reference genomes community. So what is the reference genomes community? What is ERGA? The European Reference Genome Atlas is the European node of the Earth Biogenome Project, which is the global initiative to coordinate the production of reference quality genomes for all of eukaryotic life. So in Europe, we're focused regionally on coordinating reference genome production for Europe's biodiversity. And importantly, we're also increasingly thinking about how we can foster the use of these genomics data for biodiversity protection. So on the one hand, it's building these reference libraries, but on the other, it's also thinking about how we can put these to use for biodiversity conservation. We're a bottom-up community initiatives with more than 1,000 members across Europe, with a governance structure, as you can well imagine, a sort of executive, but also a council, which is made up of country representatives, at the moment 38 different European countries. So we try to bring along all of the countries in Europe, no matter their level of current maturity in biodiversity genomics and reference genomes in particular. But basically, we focus on these 3 pillars of activity. The top one up there is focused on reference genome generation, whether that's an individual researcher in an individual lab seeking to produce a reference genome for their species of interest, or whether it's a small national project, or whether it's a large European-wide project. We also play this role as the umbrella, if you like, of all production activities that are going on in Europe, again, from the small individual labs all the way through to the big projects like the Darwin Tree of Life, where we try to connect people to make sure that they are following the same paths, if you like, wherever possible, bearing in mind things are being done at very different scales in different situations. So connecting all of the different projects in the European landscape and being that ultimate connection to the global landscape at the Earth Biogenome Project level. And finally, this kind of acting as a knowledge hub for sharing best practices, understanding what works, what doesn't work, trying to find solutions, instead of people working in silos and banging their heads against the wall because this DNA extraction doesn't work or because the assembly keeps running out of memory, etc. Try to create this forum for discussion for common problem solving, if you like. And this common forum is kind of implemented through the ERGA committees. On the top here, you have what we call mostly our scientific committees, which, if you look at the names, are basically following that production line from the sampling through to the sequencing and assembly, followed by the annotation, data analysis, and then our IT and infrastructure, which is more transversal support for all of those different kind of production value committees. But we also have kind of another perspective, thinking about ethical and legal, how we project ourselves to the public, because obviously this is public money, how we engage beyond the research community, so citizen science and other stakeholders. We try to think about that. Our Training and Knowledge Transfer Committee is also super active, and our Social Justice Committee, bearing in mind the disparities that we have in terms of infrastructures across Europe. So it's really the committees that are thinking about all of these important aspects and how we can move forward. And here I just juxtapose now the committees on the one side that you've just seen with the work packages and the 2 streams of the Biodiversity Genomics Project, to really emphasize that these 2 things have been connected all the way since the very inception. So the activities of the committee are being supported by the project. The priorities of the project are being kind of redirected by the discussions in the committees. And this is a good way of trying to ensure this continuity after a project that we were here to discuss today. So now I'll just point to a few kind of examples of project community outputs. Again, there are many, many more, but I'll just highlight a couple. And so this first one is kind of in conjunction with the Sequencing and Assembly Committee, but also with Work Package 9 from the Biodiversity Genomics Europe project. And here a very specific example of implementing a tool that allows for community assessment of the quality and basically giving it that quality control stamp at the end. This final reference genome product passes what we consider to be the metrics for the quality that we're looking at. So it's an interactive tool, and in fact, it's already being used beyond the project, but it was really driven, its development was really driven inside the project. But now community members from outside the project are also becoming engaged and acting as reviewers and increasingly submitting their own genomes for review in this common community space. So that's a really nice example of a product that has been passed on to the community. A second one is Workflow Hub, which you saw in Joanna's slides as well. And so here, the workflows are being actively developed because of the needs to deliver within the project. But of course, by placing them in Workflow Hub like this, they are now open to the community for further development and use. And here for the reference genomes, we have a suite of pipelines, workflows on assembly, and a suite of pipelines and workflows on annotation. So the key kind of production level workflows, right? A third example is this Ergo Knowledge Hub, which again has been made possible through the work of Work Package 3 in the Biodiversity Genomics Europe project, but is being fed by and used by the entire community. So it's open for, you see the big contribute button there, open for contributions by the community. When you stumble upon a training resource that you found useful, then you can submit it to the knowledge platform. It has a little quality control check, curation, and then it becomes part of the Knowledge Hub that's of use for everyone. So another example of something that is developed within the project, but in close collaboration with the community so that it can be inherited by the community and has a life after the project. A last example is the genome report setup. So this is a collection at Pensoft Rio Journal, where the technical description of how our reference genomes were produced is a very standardized report of all the input that had to go into creating this resource. And again, this is open to the community, so our community members who are not part of the project can write their project genome reports in the system and publish them all in this collection together. So giving them the tools afterwards. Sorry, now I have the final example. Again, because I always want to mention the application side of things, not just the production side of things. So here, the collaboration between Work Package 11, which is the genome applications, and our communities, where we have lots of different case studies that are kind of testing what's working and what's not working, in particular here using population genomics data to understand population structure, gene flow, hybridization, genetic composition, etc. So using these reference resources to understand the genetic health of species of concern. So finally, I've come back to this loop, if you like, with the project on the one side and the community on the other side. And I would say that this connection, if you like, brings the project legitimacy and helps to better define priorities within the project. And in the opposite direction, this connection really helps communities to advance. So we have the resources to actually move stuff forward that we want to move forward. And indeed, it attracts further participation in the community. So wherever possible, to arrange an alignment between a project and a community, and it doesn't have to be a one-to-one relationship, it could be many projects and many communities, depending on which different aspects you're looking at, is super beneficial, and we should always consider this when we are developing project proposals. So with that, well, there was a slide at the end, just to thank the Ergo community and the BGE project, and you for your attention. Thank you. Doesn't want to do that. No, there we go.
Thank you very much. I think this was a very good example of the potential of communities to really deliver a long-lasting and meaningful legacy. So I thank David for that. Now we'll be moving on to decision-making. I promised you at the beginning that we'll be exploring policy impacts for scientific results. And our colleague Damiano Odoni will be talking to us about it, specifically in the context of invasive alien species.
Yes. Hi, everybody. Thank you for all presentation before me, for all people presenting before me, because they give me a lot of inspiration inputs from my talk, especially the Quentin room, because yes, I will pick up some aspects of his presentation here. Yes, I will speak about an example of reusing the output of a project at the local level and trying to bring it up to a higher level, at the European level, where decision-making institution will play a key role. So the title explains that, from local alerts to European policy impact. as Quentin mentioned, probably you have seen the riparias in his slides as an example. Where I will speak indeed about the alert that we developed in riparias project and how we are moving thanks to Gorgeous and one-stop projects, EU-funded projects at the European level. Probably some of you know about invasive species and the problems we have with them, so I will go very fast here. Well, the first thing we have to think about, first method to avoid invasive species is prevent introduction, of course. But it's not, practically it's very difficult. So sometimes it goes wrong. So second way to prevent the spread of early invasive species, well, is early detection. So as soon you detect a species, an invasive species, less money you spend to manage it. So it's a most cost-effective way. It avoids expansion, especially you don't want this species in preserved protected areas. So I'm a research software engineer, and when I think how to have early detection, well, I think about software. I think about a software. We can call it early alert tool or early detection tool. Well, a generic software can be a web application or an app, whatever. It has to be based on data. It must to detect new occurrences. It has to trigger alerts. So people, field managers, for example, scientists, will get an email or an SMS, whatever, to know, okay, this species has been detected in this protected area, for example, or in this country, or in this region, whatever. Well, and also, who are these users? Well, as I told you now, field managers, scientists, probably policymakers, citizen scientists. It's a generic aspect. Now, in live riparias, which is a project, European project, we developed something in a project area, which is just very few river basins. The duration is 6 years. We had 30 target species. The goal of the project was the management of these species, not only the development, of course, of an early detection tool, which was the very first output of the project. So we had aquatic plant species, riparian plant species, crayfish species, in total 30 target species. 15 were established, 15 not. So we have this kind of nice way to test the tool in these two configurations. So the live riparias early alert tool is a web application. It's based on GBIF occurrences. It's an open software, and the first version was presented at TEDWIC 2022. I don't want that you remember my presentation 2022 at all. I put the links, so please. Well, but as I told, our early alert tool needs data. So the first thing that we need to do is publish this data, create repeatable workflows to publish this data. Well, thanks to Trias project mentioned, we had already this kind of pipelines. So we improved that. And not only, we actually mobilized not only the data, but also the data providers. We had a kind of an incentive for this new data publishers. We created this alert tool, which has at the moment not only the 30 species of a riparias project, we extended to 139 species, including the European Union concern list of species. We have 175 registered users at the moment, 150 alerts, more than 1,000 predefined areas, including all Natura 2000 protected areas of Belgium. And every night, there is a data import from GBIF. So as you see, 1,200, you can count how many years this tool is running. Well, but the most important stuff is that at a certain point, we thought, okay, this software is open, we are developing it, but it's really reusable because it's configured for Belgium, for a very limited list of species, how can we make this tool better for longevity? So after the end of the project, as Quentin mentioned, not only the data, but also software can get old and unsupported, unmaintained on the long term. So, well, we thought about this, and well, the target area, the species, they are just variants. What is invariant? Well, the data logic, the alert logic behind, the data visualization in some way is invariant, and the data input. So what we did is creating GBIF alert, which is the engine, which is the invariant of this tool. And all other variables, so the list of species, the list of areas, it is for Belgium, or it is for USA, or for Colombia, doesn't matter. It's just a configuration. So GBIF alert coupled with a configuration is equal to an early alert tool. Well, this was a heavy refactoring, of course, but apart to be the winner of Ebenezer Challenge of 2023, we will be able to show that this tool can be reusable in other projects. So we put this early alert tool of Riparias as an output for one-stop and Guardias projects so that this tool can be improved, but also upscaled to at the European level. We don't do this alone. We do this also, that's very important, for longevity and long life data and long life software. We do it with EASIN. And EASIN is the European Alien Species Information Network. It's the initiative of a joint research center of the European Commission, which support the implementation of the EU regulation. So actually, it's a governmental body of the European Commission. And maybe some of you know that they have this kind of NOTSIS, EASIN notification system, which is actually has a kind of official obligation. So if you put something in NOTSIS, well, you as country, member state, you must manage that species, that you put it in NOTSIS. So it's a kind of important agency, let's say, within the European Commission. So road ahead, upscaling. We have to cover all European countries. Heterogeneity of users, because we have started to make some workshops to know about this. We are the users of this, because if you go to EASIN, we have also policymakers using this early alert tool. And also alignment, maybe integration of this tool within the EASIN infrastructure, and also, indeed, the potential impact which this tool can have on decision making. But as this tool is in GBIF, as this tool, the early alert is based on GBIF data, and in 3 years we cannot make all of conversion to build this on the EASIN database, which is not technically also ready for this. Well, and also to provide to EASIN the visibility that it needs and it's worth. So we make EASIN a GBIF publisher. We push data that they collect with their app. This is an app of EASIN. We put it in GBIF. And all other data that EASIN has, we will try maybe to publish on GBIF and put it in as an EASIN network. Well, we are going now, we are just now in the phase to gather user needs via workshops. We have 1 for Guardians, and we have 2 for, or 3 maybe for workshop in the living labs of workshop to define the software requirements before the end of the year. Thank you. This is the key points, I think. Use data standards, develop good and open software. Transfer knowledge is key. And at the end of the project, try to hand over, I think before the end of the project, to hand over this knowledge. So we have collaboration with EASIN. We want at the end of the project, EASIN is part of this team of software team. Yes. That was the voilà. Thank you.
Yes.
a really great point here about how one project transitions not only into another, but a series of projects, a whole network really of collaboration. And it's truly a point to keep in mind. We'll be finishing off today looking to the future of data publishing, of academic publishing to be specific. I mentioned the ARFA publishing platform and the writing tool. Pensoft's proprietary software in this regard at the start of the session. And then I hand over to my colleague, Theodor Georgiev, to tell you all about it.
Okay, thank you, Peter. Before I start, I would like to ask you how many of you are familiar with ARFA writing tool? Okay, I didn't expect that. So one of the most important purposes of a writing tool is, of course, to facilitate the writing. And I will give you a little bit of feedback. The first version of ARFA writing tool was released 12 years ago as a part of outcome of Vibrant projects. And it was designed and developed to serve mainly for single species descriptions. Saying that, you know that this tool, it has its own limitations, and we have to think at some point to develop a new version of it. So the last couple of years we spent trying to implement all the lessons learned during these 12 years until now. So the new version of the tool, AVT 2.0, is designed as a non-domain specific writing tool. It uses, again, the same template-based documents with predefined structures, because this enables application of different validation rules. And also this structured approach is important because it serves an educational purpose, helping young researchers learn how to organize and compose scientific documents. The tool offers real-time collaboration, editing of documents. It includes also very rich functionality in terms of commenting. The other thing you can do in the new version is basically to work with different modes in like Google Doc style track change mode or suggestion mode. Okay. And additionally, the new version of the tool provides much richer functionality in terms of versioning and ability to restore the previous versions. Also, what you can do here is to import documents from XML and to export documents in XML and PDF. As I said, the tool is designed as non-domain specific, but we have implemented a lot of domain-specific things via APIs. For example, in the new version of the tool, we offer automating tagging of taxon names using global name services. Some also can be tagged manually, and all names that are marked as taxonomic names, we throw them against checklist bank and basically try to find whatever possible identifier we can, and then we store all these identifiers in the document. So when these documents are published, they have all identifiers in the document. The other thing you can do, because of this integration, you can autocomplete taxonomic names. And when you're creating treatments of already existing taxon names, you can just autocomplete, hit the name, and a big part of taxonomic treatment information is already pre-filled, like the taxon name, the authority, the hierarchical classification, the nomenclatural status, the rank. Of course, these things could be edited by the author if needed. Also, the new tool supports site ontology, which allowing all cited references to be contextualized using site ontology, which means you can say if you support or disagree with this reference, thereby this will improve the literature citation mapping. But the writing tools are not, they have not just not one purpose. They can do more. They can also facilitate the peer review process. So what you can do with the ABT 2.0, basically all collaborators or peer reviewers, they can work on one document. And they can add comments and track changes to this document. The visibility of their names, so basically if they see the policy of the journal platform, but the system enables that. And then the authors will receive a document with all the changes and suggestions at one place. And they can easily agree or disagree or improve their research papers or documents. And the other thing that you can do is basically you can compare the version that was sent to reviewers and the version which comes from the authors. And you can see if all the comments you as a reviewer left on the document are addressed by the authors. So easily can compare. This is something. ARFA writing tool is not restricted to journals only. And example for that is the collaborative solo platform. So it allows the Solve for Europe project stakeholders to contribute to the development of strategic documents called roadmaps in shared environment. So we made a custom review process for them. And those publications then can serve to address knowledge gaps relevant to the EU SOL mission and guide funding priorities. One more very important task of tools like that. Why it is important to publish data-rich documents, and here data-rich means data-rich in terms of semantic. So it's important because this allows you to disseminate the different data type to the relevant repositories. In this way, you create more fair data objects because all the different bits go to the relevant parts. But not only that, in the era of AI, tools like this contribute via embedding ontologies during writing and also linking entities to external data hubs like GBIF, like GenBank, like Catalog of Life, so adding a lot of identifiers in the documents, producing structured documents, and when published, these documents, they are immediately machine readable. So in essence, converting human readable knowledge into machine readable documents enables artificial intelligence to learn more accurately, to retrieve more precisely, and to contribute back to science. That was for me. I want to thank Bicycle Project and SOL, of course.
Thank you very much, Theodor. You can find out a lot more about ARFA at our stand, and Theodor, who's the master tech wizard of the platform, will be with us all week. So any questions go to him. I'd like to thank all our presenters. We're a little bit ahead, which is great. So everybody kept on time. I'm very happy about that, which means now we have more time for questions. I see a hand raised, yes? Just a moment. I'll just to set up the rules. I like how eager you are. Just give me a moment. I'm also mindful of the chat, but so far I think we haven't received questions from our online participants. As we only have one microphone, I'll go to you so that you can. No, I'll come to you. Don't worry, and you can ask your question. We can ask our questions first, and if there's time, I have some questions for discussion. But I'll now go to our very first question. And please let me know which presenter you're addressing it to.
Hi, Teo, there you are. My question is a simple one, I think. When you're using the global name services to find names in the publication, great. Then you mentioned the other way around, that if you have a name global names is not finding, and it's in your paper, I think that's what you're saying, you can add like the higher classification. Is that adding it to the paper for the purpose of reading, or is it actually pushing it back to global names?
So the use case is that if you're writing a paper about new species description, so obviously this name is new, not there. So in the tool, basically you can mark up this name, and you can say tag this a taxon name across the entire document. And then when you publish the document, we also issue identifier for this name. At some point, the document and the information from the document will be delivered to GBIF and catalog of life. And, yeah. So that's the idea.
Thank you. Anyone else with a question? Happy to take on. Anybody else wants to ask something? If not, I'll proceed with my. Sorry, we do have a question. Thank you, sir. Who is it to?
Actually, this is in general, but maybe since Quentin brought it up, lately there's a lot of discussion also between research infrastructures about this short-term innovation need of a lot of grants. And more there is actually plea from existing infrastructures for funders to put into their funding programs small amounts of funding for research infrastructure so that you know that certain results end up into existing research infrastructures. How does the room, and especially you, look at stuff like that?
I guess, yes, the question was directed at Quentin, but I would very much like to hear the others as well after he says it.
Yeah, well, that's a very difficult question, and perhaps I'm not really the right person. I know I can ask the question more broadly, but I would really like to sit down with our funders and talk to them more broadly about these questions, because I feel we kind of shout at each other with megaphones on two sides of a valley, and we're not really having those frank conversations about that. So I agree. I see situations where projects are competing against infrastructures, and that's not really ideal. And so I'm not going to be able to answer your question, really, but I think it's a good comment.
I'd like to hear, sorry, we have a point here, but I'd also like to hear from the other speakers on that. I just saw you first, sorry.
Just very briefly, I know on our project, we have a small amount of funds, small. They're like in the 2,000, 3,000, 4,000 range, but they are gap filling. We're looking for ways to facilitate people who want to facilitate name gaps, for example. But how do we get out to the word to different people around the world that we're in that space?
Thank you. And again, sorry that I didn't see you before.
Yeah, just to say that, so because I'm from the Swiss Institute of Bioinformatics, in Switzerland, we're actually quite far ahead with the conversation with the kind of funding agencies and the support for what we call biodata resources. So research infrastructure is just what we call them in Switzerland. So we have a very nice conversation. We have 0 results so far. While they are cutting everything everywhere, we don't even hope for some results, but we want to continue this conversation. So when the outlook is looking a little bit less gloomy than it is today, then we will maybe have some results at the end of these conversations. So yes, we need to keep having these conversations and keep developing these relationships so that they know what an infrastructure is or a resource is, and that they are connected to projects and that they are connected to communities and that they cannot be left on the sidelines.
Thank you. Yes, I'd like to hear from our co-organizers.
Yeah, as you know, the proposals are quite competitive. So dedicating a lot of our substantial part of the budget to the research infrastructures might result in a less competitive proposal, unless you ask them to join. So Catalog of Life is part of BMD, and that's a way of funding the infrastructures. And then, and on the other hand, we're building on GBIF as the global inventory repository, and that's paid by governments. Do you want to respond to that?
Yeah, I wasn't meaning that a lot of funding needs to go to that in a project. I'm just in general. If you look at, for example, European Commission, it sort of, I would say, leans on a lot of infrastructures to just magically pick up, I would say, the results at the end. And that situation, I agree, is a situation that is actually strange. So we, for example, as Catalog of Life, are also part of the Biodata Core coalition. We're talking to the funders because it's actually a weird situation. They are investing more into short-term things, but they're not investing in, how you say, the long-term longevity of the data, et cetera, So my example was only a short, I would say, reservation in projects standard, and not, I would say, not a big chunk necessarily.
Christos also has a point.
Yes, I do, because we have done this discussion on and on and on over the years. And I must remind you that the politicians have their own agendas, and they have, their own time pace at which, they want, the things to become true. They want, to achieve their own proposals, their own targets, et cetera, et cetera. So for them, I mean, 3 years is already a long way, you see? And the lesson learned actually after so many decades is how, to match the timing of the infrastructure, let's say, development, but also the maintenance in different agendas, and maintain, probably under different titles, the same kind of infrastructure that we build and we want to be there and live.
Yeah, I think that that's a good point, and that touches on what Quentin said in his presentation about this. If there's a pothole in the street, you don't build a new street, you fix the pothole. But at the same time, people who are in charge of the fundings have their own incentives. And of course, for, let's say, if we keep with the physical infrastructure metaphor here, for a local politician, it's always nicer to open up a new pedestrian bridge over a river compared to fixing potholes or something like that. And I guess the same analogy also applies to the digital infrastructure, because it's having a new shiny project, even though it's expensive, is in certain ways more attractive than like putting another round of funding to an already existing project, expanding its scope or features, or fixing issues that just turned up over time.
Thank you. Do we have any more points on this? It was really a fascinating exchange, I have to say. Anything online as well, just to be sure? Nothing? Okay, perfect. Well then, if there are any other questions, otherwise, there we go. Here you go, sir.
Andrea Tarallo, Italian CNR. This is a question for research infrastructure. I see a lot of belonging to them. Seems like that the things that we are building rely a lot on high quality data. The presentation was great. This, my point of view, is putting a lot of burden on scientists that are doing the data and depositing the data. We are asking them to do fair and open, use the right standards. So we have two ways basically, or we build easier software to incorporate easier this data, like the Alpha tool that is great, or we do invest also in data stewards. I don't see this word popping out in this conference. And we have a lot of discussion about data stewardship right now in Italy. We're trying to invest a little bit on this. I'm wondering if in research infrastructure, there is this discussion going on actually to invest on people rather than the people that have the know-how and then can help to bring this know-how along the here through the different projects, right?
A great point. I wonder if any of our speakers over here would like to respond. Please, Niels.
Yeah, so BMD heavily depends on GBIF, and in GBIF they have all these online training courses and how to mobilize your data into GBIF. So from that perspective, we built on this global infrastructure to deliver this data stewardship skills. There's a lot of online material available as well. So, and that's also why we chose for GBIF, because then that's catered for. And I don't have to take that on board in my project. So I can focus on manuals on the virtual research environments, for instance, how to operate those rather than moving the data into GBIF. That's a solution.
Please continue.
Maybe I can quickly respond. Yeah, we do have a data steward in our team. And what that person does, in actual fact, is really just connect the scientists with the people that can help them do it. They tend to not do the actual data publication themselves because otherwise they get completely overburdened. But I think it's about prioritization. It's about, yeah, scientists have only got so much time in a week, and what bits do they prioritize? And there has to be a bit of priority towards the legacy in terms of the ongoing data and software, and it tends to be traditionally that the prioritization was just get that paper out because you need the impact. And so I feel the impact comes from both. And yeah, it's trying to shift the prioritization, not go completely one way or the other.
I'm told we need to keep the microphone a little closer, but another point here. Yeah, I think yours.
I'd say to your question that there is a huge need for data stewardship in our community at all levels. The World Economic Forum put out a study in January of 2025. They basically say with better capacity development across the public and private sector, including academia, we could release trillions of dollars into the economy by 2030. So it's a ginormous need. We know it in the collection space. We have resources like Niels was pointing out, but collections will tell you they don't all have data stewards, as Quentin just pointed out. They lack the skills, or they're being asked to gain them in addition to everything else that they have. So the collections themselves need more staff. They've been crying out for this for a long time for those reasons. We want quality data to come out the door. They need people that can help them make sure that that can happen. So yeah, it's a huge topic.
Thank you. And another point over here.
Briefly introducing myself. Some people in the room might know me. I'm Stan Koolman from the Belgian Biodiversity Platform. Nowadays representing Biodiversity Officer function at INBO. But regarding that platform I referred to, I've experienced that some of my colleagues were involved in those invasive projects, but I don't completely understand why they didn't, why they weren't mentioned, or the logo or platform wasn't even included. This in the scope of this open data, not to attack someone personally, but I have just the impression that some funding might be facilitated when all parties are also mentioned and referred to.
Maybe a response? Okay, if anyone wants to.
Yeah, so I think that's a little the problem of short-term projects is that definitely the platform was involved in the TRIAS project. There were a whole bunch of partners involved in the TRIAS project, which weren't all shown in my presentation. And that project finished 3 years ago or something like this. So yeah, how do we show the contributions of all of those different people and in a succession? Because in actual fact, all of these projects, it's as Newton said, yeah, we're building on the shoulders of giants, and effectively, yeah, it's difficult to acknowledge all of those contributions.
first? Sorry. I think, yes, you. I didn't see anyone else raise their hand. Sorry if I can't catch everyone.
No, thanks very much. My name is Katrin Eil, and I'm representing Elixir, the European research infrastructure for life science data. So one thing I want to bring to the conversation when it comes to data stewardship and also data management, I think there's a lot of areas where there's still a big need to learn about those aspects, but there's also other areas where there's quite a lot of material available. And Elixir as such is also making available resources which are all open and accessible for people that want to have a low entry point to actually start building their data management plans and so on and so forth. So I'd be happy to continue the conversation. We can think of trainings that Elixir members in specific domains would be able to deliver in certain projects or so on. But what we also really do is, in a way, we have the data management expertise, but we need the people in the domains to then actually tell us what they need and to co-create those resources. So I'd be more than happy to keep chatting about this in whatever way useful.
Here, I saw you again. I think you were first.
So to that point, my name is Deborah Paul, and some of you may know me from the IDIG Bio project in the States, and now I work with the Species File Group at the University of Illinois, and we are participants in creating what you know as the Catalog of Life, global names, and also taxonworks. And I'm an active member of the Carpentries community, and I will be talking this afternoon, as a matter of fact, about this notion of a network around exactly what you just said. There's a great example I can also point to that the NFDI project, building a global sort of integrated, well, in this case it's country-based, but it can be globalized, this an integrated help desk. So thinking about how the experts from the level of the country, the level of the organization, to the level of the individual.
So it's not very common here to talk about public-private partnerships. It's like a forbidden word, especially in our community. But it's normally people think that private companies are ephemeral, so they are not sustainable. But I would say that in many cases it's rather the opposite, and can give you an example for that. We have completed as partners more than 30 EU projects historically. All those websites which were built by us are still alive. They are some 15 years after the project expirations. Why we do that? A normal institution would erase that site because it's not necessary anymore. We do that because as a private company, we understand sometimes more the value of the investment. And our own tools and workflows and publishing platforms, some of the tools have been built like the first Alpha by the Vibrant project. But between projects, we maintain and develop the tool all the time with companies' own funds taken from the market. So this is one of the ways to really think about more market-oriented approach to achieve some sustainability.
Thank you very much. Let me see if I've missed anyone. Any raised hands on these points? Damiano, please.
Just a reflection, something quite different. Just thinking about my institution, the importance to have in project proposal in this kind of projects, an institution like mine, which is a governmental institute. That means that what we do in a project, then it is applied within the policies. So we have to, as IMBO, we have to provide some reports to the ministry, and we always have a kind of a positive feedback because we use the output of TRIAS to write reports for the ministry and so on. And this generates some way longevity of the outputs, but also opportunities for new projects. So I think the importance to have governmental research institution like IMBO. I mean, I don't know in other countries, but I mean, it's great to have this kind of research institute, which is not university only, but have this kind of longevity because we are ministry based.
Thank you very much. I'm mindful of the time, and we are approaching the end of our session. Thank you for a lovely discussion. I think we raised some important points. We achieved a nice balance, I think, between prospects, opportunities, and challenges. And I'm happy to say that the session will continue tomorrow, same time, same room, and we'll have the opportunity to carry forward this conversation. I think there's a lot to learn. Tomorrow, there will be an array of completely different projects, so new insights, new perspectives, but we invite you all to participate. I think your contributions were invaluable, and I invite you now all to give applause to our lovely speakers who delivered really quality presentations. Thank you for your participation, and you can find us at the booth, and we hope to see you tomorrow as well. Thank you so much. Thank you for yours.
Okay.